So what is bioinformatics? Bioinformatics is the interdisciplinary field that develops and applies computer and computational technologies to study questions that combines biology and computer science to analyze and interpret biological data such as DNA, RNA, and protein sequences to gain insights into genetics, genomics, and related areas. So if I decipher the word bioinformatics as bio, infor, ma, t, and cs, which could be extrapolated into bio as biology, infor refers to information technology, ma as mathematics, t refers to statistics, and cs to computer science.
BLAST stands for Basic Local Alignment Search Tool and is a computer algorithm in the realm of bioinformatics. This website is accessible through the National Center for Biotechnology Information or NCBI. BLAST speeds and accuracy make it an indispensable tool in various biological research areas.
So this activity aims that the student to become proficient in using the Basic Local Alignment Search Tool or BLAST to understand the significance and idea of sequence analysis. And also, it aims to understand how to utilize the multi-align software to generate multiple sequences to identify their evolutionary relationships and common patterns.
In order to do a nucleotide sequence analysis, first, you have to access BLAST from the NCBI website. After doing so, click on Nucleotide BLAST. Enter the sequences provided in our Google Classroom, like displayed above. Input it one by one into the Enter Query sequence. Near the bottom of the page, under the Program Selection category, click BLAST. Toggle the Show Results in a new window. After a while, you will see a table showing significant matches to the sequence you search. You may explore the other feature of the database. For instance, you may see in the table the top 3 matches to your query sequence. In order to do a multiple sequence alignment, first, you have to access the online software multi-align. Enter the sequences one by one in the query box, separating each sequence by pressing Enter. Click the Start Multi-align button and wait for the software to analyze your data. Once it is done, you may edit the image in Paint, highlighting all the sequences that can be used to differentiate the sequences. From this data, you can now proceed to make an analysis.
For each accession, what are the top 3 matches to your sequence? So for the M1, M2, M3 DNA sequence data, the first one is Ipomea tenissima, the second one is Ipomea lacunosa, the third one is Ipomea batatas. So for the second guide question, what percent identity of each aligned sequence showed gaps indicating sequence differences? So as shown on the table, the M1 DNA sequence data showed 2% gap, showing the highest sequence gap among all the data. So for the last guide question from part 1, from what species the first match was derived from, and what is its gene function? So for the M1 rev-revcom and M2 rev-revcom, this involves Ipomea tivita, which is subjected to pairwise alignment.
The top 3 matches in the query sequence compared to the other DNA alignment sequences are typically defined because they have the highest sequence similarity scores. This means that they have the fewest mismatches and gaps when aligned with the query sequence. For example, suppose we are interested in finding the closest relatives to the gene M14. The top 3 matches in the query sequence are likely to be the closest relatives to the self gene. We can then use this information to study the evolution of M14 and to identify the other genes that may be involved in the similar biological processes.
Moving on with the part to the multiple sequence alignment. The analysis of the first 80 to 90 base pairs of the sequences. The first 80 to 90 base pairs of the sequences in the sample have incomplete DNA alignment, which is necessary to generate a complete multiple sequence alignment. M14 and M34 complete lactate DNA alignment from 1 to 89 base pairs. This base pair also contains conserved sequences, as can be seen in the lowest part of the multiple sequence alignment. The blue color from the base pairs before 90 represents a weakly conserved residue. Moreover, the consensus line, represented by a dot, shows a position with no conserved residue. The 8 to 398 base pairs show conserved sequences for all entries, which are identical or similar sequences in the alignment. One informative character present in the multiple sequence alignment is the deletion events, which are indicated by the presence of gaps or hyphens in one or more sequences. Lastly, substitution events also take place, which are indicated by differences in characters at a specific position in one or more sequences.
3 colors, the results show that there are 3 types of mutation present in DNA sequence, 52 deletion events, 23 insertion, and 20 substitutions. According to Gupta et al., sequence analysis helps us to detect evolutionary relationship as well as scan motifs by taking into consideration of various events such as mutations, insertions, deletions, and their ordering under some circumstances. A mutation is a persistent genetic change that is heritable and can cause phenotypic changes as well as change protein function.
Bioinformatics' capacity to manage high-throughput data is a major contribution. Researchers may now create gigabytes of genetic data in one experiment because of DNA sequencing advances. Through the help of BLAST, we find regions of similarity between biological sequences. The program compares nucleotides or protein sequences to sequences, databases, and calculates statistical significance.
Thus, the statistical methods of analysis in bioinformatics provide a relevant use not only in a certain complex crop species, but also in modern biology and medicine, showing the importance of computer and answering thousands and millions of questions and possibilities in biological data, and to further contribute to the future human genome.
Protein sequence analysis. Bioinformatics is extensively used to analyze and compare protein sequences, predict their structures, and understand their functions, helping to understand complex interactions and networks within cells, tissues, and organisms. Evolutionary biology. Bioinformatics tools are used to reconstruct phylogenetic trees and analyze molecular data to study the evolution of species and genes. Structural variations and mutations. Bioinformatics aids in the identification of structural variations and mutations in the genome, which can be associated with diseases or traits. Environmental genomics. Bioinformatics is used in the environmental studies to analyze genetic information from ecosystems, aiding in biodiversity assessments and understanding environmental changes. These are just a few examples of many applications of bioinformatics in the biological and biomedical sciences. The field continues to evolve and expand as new data and technologies become available.